Alright, so in this video we are going to be talking about one particular group of organic compounds, our organic macromolecules, and how they play a role as enzymes and catalysts in reactions. So, the first thing to start out with is, what is a catalyst? Before we figure out what organic molecules we're talking about, a catalyst is going to be a molecule or some sort of chemical that speeds up the rate of a chemical reaction by lowering the activation energy. So activation energy is the energy that it's needed to start a chemical reaction. We can think of this as like putting a coin in to start an arcade game, or the push needed to roll a rock down a hill. So these catalysts are going to cause the rate of reaction to be faster, because they are reducing the amount that you have to put into the actual reaction. We see this a lot. We see this in our everyday lives in catalytic converters. Catalytic converters use a couple different metals that will convert the toxic carbon monoxide and nitrogen oxide from your car's exhaust and convert it into a slightly less harmful carbon dioxide and nitrogen gas. So this reaction can happen naturally, but thanks to the catalysts in the catalytic converter, it happens a lot faster to make our cars safer. Other catalysts you will see happen in your body. All of the catalysts in your body are set to keep the reactions in your body happening at 98.6 degrees Fahrenheit, about body temperature, about average body temperature. So these catalysts allow all of the reactions to happen in your body at a much faster rate, which helps to keep you functioning. So if we look at a reaction diagram, we can see that without an enzyme, there is a big amount of energy needed to start this reaction. So again, that push to get this reaction to start happening. As you can see, it ends with the production of carbon dioxide and water. If we add an enzyme, that amount of energy needed is reduced significantly. So the hill that we have to cross to get this reaction started is going to be much lower with the enzyme. The end product is still the same, regardless of whether you're using an enzyme or not. The enzyme or the catalyst is just here to make it easier to do the reaction. So that leads us into enzymes. So if you'd like, there are some other videos on the notes that you can watch to kind of boost your knowledge of the topic. So this talks about enzymes, what they are and what they do. But I am going to move on and kind of talk about what they are outside of the video. So enzymes are proteins. Proteins, we said, are used for enzymes. They're used for structural material. They're used for muscles. But in this case, we are going to be talking about specialized proteins that speed up that rate of reaction by lowering the activation energy. So this whole part here, again, these are catalysts. They are going to speed up reactions by lowering that push, that energy needed to start the reaction. You can see down here, we have our enzyme, and it has this special spot on it. That spot fits what we call the substrate. The substrate is just going to be the chemical, the molecules that are going to be acted on by the enzyme. And this enzyme is going to react these two molecules together. It's not going to actually take part in the reaction, but it's going to cause these two molecules to react, and it's going to produce this product. Now in this case, you can see that it breaks them apart. We can have reactions that form them back together. So there are enzymes that put things together just as much as there are ones that break things apart. Now this spot that you see, it's called the active site. And the active site is the location on the enzyme where the substrate is going to bind and react in. So you can see on this enzyme here, we have a very specific shaped active site that fits this very specific substrate. So when the substrate binds, it fits into the little grooves there, the reaction happens, and the products are spit back out. So with that, enzymes are specific. So the model we usually use to think of this is called the lock and key model. And the way we think of it is that enzymes, in order to be very efficient, they need to focus on one type of chemical, one molecule. So for each enzyme, there is one specific substrate that is going to fit in the active site. So for example, if I draw a different substrate that is a circle shape, it can't fit into that active site. So the key does not fit into the lock in this case. The reason that this is because we can't have the enzyme stuck doing 20 different tasks at the same time. It's not efficient. So you end up getting an enzyme that without maybe one of these different molecules, it can't function. It can't work at the same time. And what that does is that leads them to be less efficient, and the organism or the thing that needs that enzyme is not going to be able to survive. So by focusing on one substrate, we can focus all of our efforts, we can do things a lot faster, a lot more efficiently, and we can overall have a better use for the enzyme. The other thing about enzymes is that they are reusable. So like I said, enzymes don't actually play a part in the reaction. They are here to facilitate, to help the reaction along. So enzymes are reused over and over and over again. Enzymes can actually be used thousands of times to catalyze thousands of different reactions. So you can see here we have the substrate binds the active site to form this complex here. The reaction occurs, we spit out the product, and then the enzyme, not having anything done to it, can go back to do another reaction. The only time you are going to get an enzyme that stops working is when it denatures. We're going to talk about what denaturing means here in a little bit. But enzymes don't play a role in the actual reaction. They just help to reduce the energy so that reaction happens faster. It gives them a place to react. Okay, so that's going to lead us into enzyme function. So enzymes, even though they are really good at their job, they are really picky. And what that means, there are a couple different factors in the environment that is going to affect how well an enzyme is going to work. The first one is temperature. So temperature is really important to enzymes because they like a very specific range to work in. If they are too cold, what happens is the reaction doesn't happen as fast and the molecules aren't going to be broken up as quickly. As we raise the temperature, the reaction is going to get faster and faster until we hit a point where we get these extreme temperatures that are going to cause the enzyme to stop working. It's going to lose its shape and become denatured. So you can see in the diagram over here, we have this very specific shape for the active site. If we raise the temperature and we cause it to denature, it loses that shape, so it can't fit anymore. We actually just saw this in class. We talked about catalase found in potatoes and how over our lab, if we heated the catalase enzyme on the hot plate and boiled it, the enzyme actually stopped working. All of our data was showing that there was no activity from the enzymes because we essentially just cooked them. We made them lose that shape, which means that they weren't able to catalyze the reaction. So looking at a diagram here, we have four different types of four different enzymes. We have one found at 4 degrees. 4 degrees is almost freezing. We'd say that's probably about maybe 8 or 10 degrees Fahrenheit. 37 is about room temperature, and 95 degrees is almost boiling, so maybe about 200 degrees Fahrenheit. And when we're looking at this, So we can see that this enzyme here works really well near freezing. We see that this is actually the enzyme for an Alaskan shrimp, or an enzyme found in Alaskan shrimp. And as we get into higher temperatures, you see that this rate of reaction drops and kind of flattens. This is when the denaturing is going to occur. So at this point, the reaction isn't really happening. There may be a little bit here and there, but overall, the reaction isn't happening. The enzyme, if it was shaped like that, has lost its shape and now does not work properly. If we were to take these enzymes found in humans at 37 degrees Celsius, and we were to put them in about boiling temperatures, the same thing happens. We lose the ability to perform that reaction. The enzyme denatures, and we can't do it anymore. Up on the right here, we have the enzymes from a bacteria found in hot springs, so very, very hot conditions. So if we were to put that into more room temperature conditions, you can see that they're not going to work really well. And if we take it any higher than boiling point, it's also going to lose its ability very quickly. The next environmental condition is pH, and pH is the measure of acidity or basicness of a solution. So each enzyme has its own special pH. For example, the enzymes found in your stomach work best at a pH of about 2 because of all the stomach acid. So if we change the pH outside of their happy range, again, it's going to cause them to lose shape and to denature. So we can see this in these 4 enzymes here. You can see chymotrypsin, cholesterinase, pepsin, and papin. Chymotrypsin works best at a pH of about 8. If we take it outside of that range, you can see that the activity falls to 0 on both sides. Cholesterinase, on the other hand, likes it really basic. So you can see at about 7, it starts working really well, and as we get more and more basic, the enzyme retains that high activity. On the other hand, pepsin is found in your stomach, which makes sense that it has a very acidic pH. And as we take it into more basic, more neutral ranges, it's going to stop working. Papin, on the other hand, works best at pretty much any condition. We can see, we can't tell what's happening past 4 or past 8, but in this range, it's going to work well no matter what conditions it's in. So you can see that even though they all have specific ranges, those ranges can vary between different enzymes. Right. The last 2 conditions we're going to talk about are concentration. We're going to talk about enzyme concentration and substrate concentration. Enzyme concentration affects the ability because the more enzyme we have, the better the reaction is going to happen, the faster it's going to happen. I like to think of this as giving my students homework or giving them an assignment. If we have one student working on a 10-page paper, it may take them a day, maybe 2 days to get done. If I give that same assignment to 5 or 10 students, it's going to take significantly less time to happen. They're going to get it done a whole lot faster. In the case of enzymes, the more enzyme we have, the better the reaction is going to go. The more people working on the project, so the faster it's going to go. Substrate, on the other hand, has a limit to it. So the more substrate there is, the higher the activity is going to be, the faster that the enzymes can grab a new molecule, react it, and then move on to the next one. There comes an issue, though, when we get to a point where the enzyme can't handle any more substrate. So if all of the enzymes are working, then we hit what's called the point of saturation. The point of saturation is this kind of plateau here you can see on the chart. The enzyme can't work faster to get all of these done. So if we can keep the amount of enzyme consistent, they get to a point where they can't work any faster and they can't do the reaction any faster. So again, this is what we refer to as the point of saturation. You can just see, comparing these two up close, enzyme concentration is very linear. You have more enzyme concentration is going to make things happen faster. In substrate concentration, however, again, we have this growth as we add more and more substrate. It's going to react faster to a point where we can't do any better. We can't work any faster, which means that we are going to hit this point of saturation. So this is where we are going to end for today. This will be the last video of this unit. So in the next unit, we will be moving on to cell anatomy, cell parts, and how we identify cells using microscopy and things like that. So if you have any questions, feel free to email me, and I hope to see you in class.